Welcome to the Maine Science Podcast. I'm Kate Dickerson. Today's episode is a conversation with Heather Hamlin, who is an associate professor in marine science at the University of Maine. I've known Heather for years, and she's been part of the Maine Science Festival in multiple ways. Heather gave a really great talk at our 2017 5-Minute Genius event, and she was one of the scientists that composer Lucas Richmond spoke with as part of his research for the Warning Sea. Heather's commitment to science, research, her students, and Maine come through loud and clear in our conversation. I hope you enjoy it. Welcome to the Maine Science Podcast. I know you're at the University of Maine, and I know you're a native Mainer, which I was going to say I'm jealous, but I'm not jealous. I just think that's really cool. I always love the stories where folks who grew up here and want to be here get to come back.
So obviously I started here, as an embryo. This is where I was. And growing up in Maine, I loved it, but I was so eager to experience more. And so I did that, but in fact, I did my undergrad at the University of Maine and then my master's. And then I was like, okay, I feel like I need to experience more than just this sort of siloed world that I've been in for everything, not just for my, but for personal growth, but for professional growth. And at the time I was, economically, I was like, oh, I don't know what will happen with me. Who knows, right? And so I think I should experience these things. So I did. And I went to, I think, New Jersey first and worked for the National Marine Fisheries Service. And that was fantastic. And then I went to Florida. And obviously in Florida, it was sort of the warm water oceans mecca because, here I was, a marine scientist, and it was fantastic. And I found this lab, Mote Marine Lab, which is like the National Center for Shark Research, and they do all kinds of these great things. And they had this fantastic aquaculture park. And so professionally, I really blossomed. But what ended up happening along the way is I met who would eventually be the most amazing mentor and completely shaped my life and my career. And I kind of found him serendipitously. I was interested in this contaminant nitrate that there really wasn't that much research on at the time. And someone happened to mention in a meeting that this person at the university, this guy Lou, was working on this. And so me being naive, right? So I reach out to him and I'm like, hi, my name's Heather. And I think this thing nitrate might be kind of important, and so he emailed me back, which was great. I didn't really think anything about it at the time. And so he emailed me back and said, yeah, we should talk. Scheduled a meeting, right? Talked with him. And to make a very long story short, I think being naive was the best way to go, because it turns out he was a huge deal, like a really big deal. Like in fact, later he got the Heinz Award for which is like the Nobel Prize for the environment, and he was this really big deal, which I never really knew until a couple of years after I was working with him. And so, but the take home is he really took me under his wing and he brought me into this science world of really amazing people, like really encouraging, really amazing people. But he also taught me science at a level that was just, mind blowing. I look back and I'm like, this is incredible. And so from there, this was in Florida and then I ended up at the Medical University of South Carolina. But along the way, I started to get really homesick, even though I was experiencing these things, and it was fantastic. And I wouldn't change that for the world. There's something about our region and Maine that is home, and I appreciated it more after leaving, right? Because all of those things, you're like, oh, I want to get out and experience the world, and you do. But wow, Maine is an amazing place. It really is. And I came here for a conference and it was just so homesick at that point because you remember, all of the things. And so then it really became my goal to get back here. And I am still sort of gobsmacked at how that happened, because finding jobs in academia is really difficult, and finding jobs where you want them is almost unheard of, right? So it's just, I don't know, serendipitous. This job came up, at the right time and sort of the right scenario. And I applied. And so I ended up at the School of Marine Sciences through the Aquaculture Research Institute. And it's been, and it's really been fantastic ever since. It's been a great group. Maine is exactly what I hoped it would be.
You said nitriles, right?
Nitrite.
Which is not at all what I would think of when I think of marine science. How did they weave together? Because you said Florida too is very different, right? So is that another aspect of it, or is that specific to this as well?
So so nitrate, with that, I started becoming interested in aquaculture while I was in Maine. And at the time it was very pie in the sky. I wanted to feed the world. And I thought that's how you could feed the world. And I'm still convinced you can in a much more environmentally friendly way. Right. And so, seafood is really important. And so I started, this was many, many years ago, way more than a decade ago at this point. And so I decided that I wanted to focus on something called recirculating aquaculture systems. And this is a long time ago. So it certainly wasn't that common in sort of commercial applications at that point. But I thought in the future that would be a way to grow fish in a really ecologically sustainable way. And so, but one of the limiting factors for growing fish in systems like that was this thing called nitrate, because it builds in the system over time. And the engineers are really good at developing filtration systems that will get rid of most of the other stuff, right? But the filtration systems that get rid of nitrate are really technically challenging and complex and expensive. So what farmers usually do is use water exchange. Well, water is now becoming so much more valuable. And so, and so that's sort of its own commodity. And in that sense, so I was like, okay, we need to figure out, what concentrations of nitrate are even acceptable, because at the time the thought was, oh, it's fine. They'll be fine. Right. You know what I mean? As long as it doesn't kill them. And I'm like, I don't know if that's true. Turns out it's not true, and so that's how I started. But then when I met the mentor, right, who's working on this, he's like, yeah, it's also an environmental concern. And it turns out it's the second leading global pollutant next to CO2. And so, right, yeah, it's tremendously impactful. In fact, think about everything that's made of like nitrogen, right? And nitrogenous waste, and trees break down and animals break down. And, it's this huge process. Well, we've more than doubled basically the nitrogen input to the environment. And so it's a big deal. It's a really big deal.
You were one of my go-to scientists when I took Lucas Richmond on a tour of the Gulf of Maine researchers and practitioners. And you were talking to him about lobster. And I have since seen that was published. So maybe you could talk about why you moved into lobster when you got to Maine.
Yeah. So I mean, how can you not? I guess is really is the easy answer in Maine. So yeah. So when I moved to Maine, obviously, Maine is important to me. The region is important to me. So I wanted to make sure that I focused on. I've always been interested in economically important animals, right? That's part of the whole aquaculture premise. But when I came to Maine, environmentally friendly, practices, but economically important animals in Maine extends to beyond animals that we just culture and grow, right? So the fishery in Maine, lobsters is such an iconic part of our state. And with the environment changing so rapidly, I'm still interested in, say, Atlantic salmon culture. I still do research with that. But I think I would be really remiss not to also focus on lobsters, which are so, economically important, but they're a part of our heritage. And the more I learned about sort of lobsters, and even though it's booming right now, and I hope it continues to do that, right? But I think we should be aware of what the potential consequences are for environmental change. And if that means we find out, wow, I think we're going to be safe, that's fantastic. That's great. But the more we know, the more informed we can be. And I think the lobstering community wants that too. I think they want to know, okay, what could potentially happen? How worried should we be? Those kinds of things. I just think the more we know, the better it will be.
When you say environmental change, I'm assuming, maybe I shouldn't, that you're talking about climate change specifically in the Gulf of Maine.
So there's so many things that are happening with the environment, but one of obviously the most impactful right now is both temperature and ocean acidification. And especially for Maine, we are more, vulnerable in some ways to increasing ocean temperatures because our region, for a number of reasons, the temperatures here are increasing at a rate that's greater than most other places in the world. And so because of that, and because of that change that's, happening more here, I think we are particularly vulnerable, or we could be, if depending on, the resiliency of the animals that we have here, which we're still trying to figure out what that means, and how resilient they are.
It's not just the warming, it's ocean acidification, which maybe you could explain how that happens and then how that impacts lobsters in particular.
Yeah, so how it happens is actually really sort of easy to explain in the sense that just like if I took a straw, right? So we exhale, right? And we produce carbon dioxide, right? And so there's so many processes that also do that, cars, and there's so many things in our environment that produces carbon dioxide. And if I took a straw and I blew into water and bubbled, blub, 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 right? The pH in that water drops just because of that's, basically how it naturally interacts with the CO2, and it increases hydrogen ions and all this stuff. But basically the pH goes down.
And when it goes down, it gets more acidic.
It becomes more acidic. Yep, exactly. And so we're in a bubble, right? The Earth is in this atmospheric bubble, and we're all breathing into that bubble. And that bubble, which is great, you need that, right? You need that layer to hold it in. But what's happening is it's trapping it, right? And so the CO2 in the environment is just going up and up and up. And so it's funny, we run these trials, right? These ocean acidification trials in a room that has these tanks that even though they're sealed, you know what I mean? Even though they're relatively sealed, they monitor the CO2 level in that airspace between the water and where it's sealed, because that's how we change the pH, right? We add CO2 and then we change the pH and make it more acidic, just like is happening in the world. But it really resonates with me because when we walk in that room, the sensors know it. Just from my walking in the room and the breath, my breathing, the whole room senses it, right? And it can change the pH in that airspace. And just so it really resonates with me that, yeah, wow, it's very easy for us to have an impact on the whole, world, basically, because of the amount of CO2 that we're producing. And CO2 is actually dissolves easier into water than oxygen does, which is not great for ocean acidification and climate change. But yes, yeah.
One of the things I got to learn when I eavesdropped on your conversation with Lucas is that you don't just scoop up ocean water or Gulf of Maine water and use it in your experiments. You design your own water and you just explained why, right? So you can change the pH. So what do you see when you do change the acidification or you change the temperature? What happens to those critters that you're watching?
Yeah, so I guess, so other studies, one of the things we were looking at is other studies basically said that, yes, things like bivalves, so like oysters, right? Things with two shells, those kinds of animals are really sensitive. But lobsters are less sensitive because they have this great ability to what we call buffer their internal concentration. So even though the water is acidic, they can sort of buffer that inside and maintain what would be ideal for them. And so in that sense, we probably don't have to worry. But what my research group is thinking, they can do that, but that takes a ton of energy, right? It takes a lot of energy to do that. And if they're putting their energy into combating that acidification, does that mean that it's reducing their bandwidth to deal with other stuff, right? Like, okay, what if now the temperature changes? That's a whole nother stressor. What if they get hit with a disease, right? A pathogen? They're going to have to fight that too. And you've only got so much energy to deal with all this stuff. So one of the things that we wanted to do is understand, well, okay, does that change their capacity to deal with some kind of subsequent stress event? So the first thing we wanted to do is sort of stress them out and see if the ones that were exposed to ocean acidification sort of got more stressed, right, faster than the ones that didn't. Did they respond the same, I guess, to those stressors? And I guess you could liken it if you were, well, you are a person, but if you take people and put them on a treadmill, right? And so you might think, okay, the people who don't have as much bandwidth to deal with, whatever else might fall off the treadmill faster as we increase the speed. So what we were trying to do is something similar. And so what we did is slowly put them in a temperature ramp. So we increase the temperature, right, slowly. As you do that with animals like lobsters that are sort of vulnerable to temperature change, like we always maintain the same internal temperature, well, there's changes as we increase it. And so their heart rate goes up as we increase the temperature. And it goes up and it goes up and it goes up as we increase it until there's a point where you're kind of hitting their max. And then their heart rate, and this is not lethal, by the way, this is just like normal, right? And so until it starts to change and then it gets a little bit erratic and that will actually go down, right? Yes, if you continue to do that, you would probably kill them, but that wasn't our intention. In fact, all these lobsters, we just put them back in regular water and they survived and everything was fine. But that point at which it gets sort of erratic, their heart rate, that's called the Arrhenius break temperature. So that's the point at which they're physiologically unhappy, right? So their heart rate tells you I broke right here. And so at this very exact temperature. And so, yeah, we use some fancy modeling to figure out exactly where that temperature is. But the cool thing is what we found is that those lobsters exposed to the more acidic water. And so we used other scientists' models that showed what the pH will be in, say, 50 years. So we're like, okay, let's project out. This is what they're predicting the ocean pH will look like at that point. That's what we'll use as our future projected pH and for acidification. And we found that the lobsters exposed to that actually sort of fell off the treadmill faster, right? They broke sooner than the lobsters that were exposed to current condition acidification and pH.
So that's a bummer.
It is a bummer, but to me, the bigger bummer is we followed that up later with the sort of the so what. Okay, so maybe they do have reduced bandwidth, but let's make that a little bit more meaningful. And we actually followed up with a couple of really cool studies. We repeated it. And so showing that if we then take those lobsters and expose them to a pathogen, right? Something called gefchemia. And so it's one of the diseases that used to be more of a problem in lobsters, like an impoundment. And so if we expose them to that the lobsters who were previously exposed to ocean acidification, not even at the time, right? They've had time to recover. That the ones that were previously exposed to ocean acidification actually died sooner than the ones that weren't. So that really is the so what of, okay, so yeah, it does look like they do have a reduced bandwidth to deal with subsequent stress, and disease is one of those stressors that they might be more vulnerable to.
So is the end goal for this, I know you said you're interested in economically important aquaculture species. Is the end goal for this is to, well, what is the end goal? I'm not going to put words in your mouth.
Yeah. So, well, it's really to understand, okay, so if our populations might be, and we don't know, there's so much that happens in the real world that could be different than our microcosm of the lab environment. But if that is the case, and this does hold true for environmental conditions outside of this, so real world conditions, then I think we have to be really diligent about making sure that we communicate with, like, say, Department of Marine Resources, who are doing trawl surveys, that they're being really, which they are anyway, but really paying attention to the potential for disease. So if they know that, studies are showing that they are probably more vulnerable, especially as things like acidification get worse and maybe temperature, right, continues to increase, that everybody will want to be more vigilant about watching for things like disease, including lobstermen. And maybe being more readily reporting, like, I'm seeing more of X, Y, or Z, because I think lobstermen are actually some of the sort of the initial wave of their doing it all the time, and yes, DMR certainly does trawl surveys, et cetera, but they have how many boats, right? When, the lobstering community has how many boats. So I think working together, or something like that would be really important.
Do you ever get any pushback from either DMR or lobstermen who are saying, yeah, but that's not real world conditions? Or do they understand that you're trying to project into the future a certain amount so that you can see what might happen?
Well, I personally haven't, but I could see where it could happen. And I've seen, though, I think the problem lies if we did this study and then we went to the media and was, oh my God, our lobsters are going to, perish. And, and then that would be a problem because we don't know that. And I think that's probably happened in the past. And I think that could potentially negatively impact the lobstering community, right? So I think as long as, we are realistic in our findings and we let people know that doesn't mean, right, that our lobster populations are going to crash. It just means that we need to pay attention. I think that's okay. I think as long as we don't overreach, then trust is maintained on both sides. And I think that's really kind of the important point.
Do you have any other species you're working on other than Atlantic salmon and lobster? And I'm going to make this a twofold question. Do you have students who come to you with, I have a great idea, and you kind of let them run with it?
Sometimes. But what we do is sometimes we'll be like, we have a great idea. Do you want to learn how to do science? In addition to caring about ocean acidification and temperature and those kinds of things, and obviously nitrates, right? So that's important. We also, because of this region, also care about something called arsenic. And so in New England and in other parts of the world, arsenic is really high naturally, but then we do all kinds of things to the environment to increase that even more. We include it in pesticides and things like that, so it can become even higher. But because it's really important here, and because other places in the world have shown that it's high, we were kind of interested in understanding what impacts arsenic might have on aquatic animals. So this is actually, I think this is really kind of so we brought undergrads in to help us, to work on this. And we actually used, bettas, Siamese fighting fish, like the gaudy, with really long fins that people put in a cup, right? That actually turned out to be an amazing model to understand behavioral changes, because behavior in a fish can be kind of hard to quantify, right? It's like, I don't know, it's just swimming, right? But bettas have these really cool stereotypic, what we call, so these repeated behaviors, like when they're mad, they flare out their gills, they erect their fins, they bite. So you can really kind of quantify, I guess, right? The number of times it does this. And I've become increasingly interested in behavior because as a physiologist, you see all of these, what we call endpoints, right? Okay, there's heart rate, or there's this blood chemistry parameter that goes up or down or whatever, but there's always this sort of so what factor. Like, what does that mean? Is it really going to change the population? Is it really harmful? Behavior is one of those really what we call downstream effects that if something is affected, because it's the accumulation, cumulative, sort of everything comes together to create behavior. And so that's why I've been really interested in that sort of downstream so what effect. And then I'm looking at that. And then if I find something, I'll work backwards then, right? I'll go backwards and see what caused it. What are the mechanisms? So for arsenic, this is really cool. So what we found with arsenic is even at really low, we call them ecologically relevant concentrations. So that means concentrations that are high, but you could find them in the environment, meaning it's not rare to see them at these high concentrations. And so the other cool thing about bettas, you've seen them, right? They come in different colors. And so naturally, female bettas prefer red males. Okay, that's just, it's a thing, right? And so other studies have shown that. And we repeated that and they do. And so what we have shown is, and you can tell they prefer them because they spend more time near them. And that's sort of a proxy for, I'm choosing you, right? Other studies have shown that, right? So I'm choosing you. And what we found is that normally, yes, they chose the red males, except when they were exposed to really low concentrations of arsenic, they didn't choose anything anymore. They didn't choose red, they didn't choose blue, they just lost their preference. And why that's important, especially important in betas. So that, that red color that you see, well, that is actually from their diet. So those are called carotenoid pigments, and they get that from their diet. So unlike the blue, which you don't change with diet, the red, the more they eat those carotenoid pigments, the redder they become. So it's what we call an honest signal of foraging ability, or the ability to go and find food. So in that sense, you can kind of understand that, oh, so the female, I could see why females, like, I kind of like the blue, but I'm not a beta. And so I can see where, right, if you're like, oh, if this one is really, really red, it's a good forager, right? It's good at finding food. The other thing is those pigments do more than are just pretty. Those carotenoid pigments also are involved in immune function. So if somebody's really red, that means that they've satisfied enough of that carotenoid for their immune system, and they're allocating some for being pretty. So there's some real benefits to choosing red. And so if all of a sudden those preferences just go away, what could that mean, right? That could really have sort of population level effects. So then, in fact, so Scarlett Tudor, right? But, she's a behaviorist. And so she's like, but I wonder why. And so she's like, stress and anxiety can change these preferences and can make us behave in ways that are maladaptive, that are not beneficial. So in fishes, one of the ways that you can sort of assess that is it's called scototaxis. So it's basically you put them in this tank where they have an option to go in the dark area or the option to go in the light area. And fishes, that's been verified with pharmaceuticals and some other things, that fishes that go in the dark area are more stressed. They have higher anxiety. Versus the ones that go in the light area spend equal time in both. That's normal behavior. I'm not stressed. I'm going in the light area to maybe look for food, right? It's a sort of a metric of boldness, like, hi, you know what I mean? Versus I'm going to also explore this side. So just kind of going in both places. And sure enough, right, the betas that were exposed to the arsenic spent significantly more time in the dark section. And then some fancy modeling, looking at correlations between the two, right? You can see that the ones that had a stronger preference for red were the least anxious. And then, it kind of goes through. So it does look like it was that anxiety, right? And that stress that was facilitating the choice, or lack thereof, I guess, in that case.
There are so many questions that I have with this.
And does that happen with people? Potentially.
Part of me wonders why there'd ever be blue at all if it's not beneficial for mating, but that's just a total diversion.
Yeah, no. And that's actually a really interesting question. So there's probably some keep in mind that the blue is not like what you find in the natural environment is very different than what we have bred. So, right. And so, so that's a really difficult question. So do you find those really vibrant, bright blue betas in the wild? I don't know. That's it. I really don't know the answer to that.
Oh, good. I stumped you. And then and the other one, the arsenic, I assume what happens is it comes from rainwater or runoff.
Oh.
Because it's in the, I know it's often found in rock and granite and things like that. How does it end up in the water system?
Yeah, through leaching, just like what you said. That's a great point. Yeah. So it basically leaches out of bedrock, or it can end up in groundwater from the intentional application of fertilizers that contain arsenic or something like that. Yep, exactly. So it basically leaches from natural formations mostly.
And is there any way to filter it out?
Absolutely. Yeah. I guess arson, and I say I guess because this isn't my bailiwick, but
I know I'm totally putting you on the spot. I'm just curious at this point.
But we have a filter, right? That we got a special extra filter that also filters out metalloids. Like that's what arsenic is. It's a metalloid. It's, and so it's not quite the same as a heavy metal, but so it filters those out. But if you have like household filters, that some of them will specifically filter out things like arsenic as well.
And that would be important in places like Maine where there is such a high natural occurrence of it, right?
Very much, even on well water, right? Especially well water. So that's a good reason for people in Maine to get their wells tested on a pretty regular basis.
So what are next steps for projects for you?
Yeah, so there's a couple of things. One, I definitely want to continue on with the lobster research because there's still so many unanswered questions about a number of things. But one of the questions that we have stems from contaminants in the environment and how those might impact lobster survival, lobster behavior, right? Things like that. So we want to sort of extend that to understand how contaminants in the environment might affect lobsters, in addition to continuing on with understanding temperature, acidification effects.
Before you go on, what do you define as contaminants?
It could be pesticides. It could be, there's so many things, pharmaceuticals, there's so many things that end up in the water that unfortunately we have a lot to choose from.
Okay, so contaminants, which if I remember correctly, you have been studying environmental contaminants from pharmaceuticals in particular for a while. Isn't that what you talked about? And I'm going to put you on the spot again in that 5-Minute Genius talk from way back.
Yeah.
About pharmaceuticals in particular, because it was I remember it because it was not something I had thought about much before that.
Yeah. And so, and funny that is actually one of the mechanisms, right, that can actually change behaviors and change stress patterns and things like that. So so I have been interested certainly in that for a while, and I'm not exactly sure what contaminants we'll end up, potentially looking at. But that's really one sort of area of investigation that we're moving toward, and I'm really excited about. And then the other is, so as I'm sure you've heard, we have now, which is like my world is coming back around, and I'm so excited because we have these recirculating aquaculture farms, right, that are coming to Maine. And this wasn't even on my radar when I came here. So now I am like beyond elated, right, that this is happening. So I really want to get back to looking at recirculating aquaculture systems and things like nitrate and how that can impact the animals that live in it, because that's what I was interested in from right at the very beginning, because that's how you're going to feed the world, right? In a really environmentally. I'm excited about that. And that's an area that I really want to start to focus more heavily on.
This is where you have graduate students and undergrads and postdocs jump on the team and help out as well, right? So you can piece together some of these different projects.
Oh, with all of them. I mean, there's no way I could do any of this without students, right? I mean, it just the one, I don't have the bandwidth, but two, that's part of the whole, my job, right, is to train, right, the next sort of generation of scientists. And I love that part. Plus you get to form, scientific communities and, and these are your, so back to Lou, right, my mentor, he was like, the students, that is your sort of your legacy. Right. What you do is one thing, but your legacy really are your students. And so for me, that's probably the most important thing I do, is working with students. And like I said, without students and without colleagues and without collaborations, you really can't do much of this. So
one last thing I'll ask you about, as someone who, I have a degree in chemistry. So I spent a summer in a lab, which is where I learned that was not going to be my one of the best things I ever did was learn that I loved chemistry, the lab work, not so much. But I don't remember a lot of people in my undergrad getting into labs before junior or senior year. And my sense has always been that is about the exact opposite at UMaine, that there is a huge opportunity for students with no experience or very little experience to jump in with researchers who will help guide them through. Am I correct on that? Because the reason I bring this up is because you said that's the most important part of your job. And I've spoken with enough faculty to know that, it doesn't seem to be restricted. And I just want to clarify that or make sure that I understand it right.
You absolutely do. Is it more common for juniors and seniors to be in labs than it is for, say, freshmen and sophomores? Yes, but I think that's in part because as undergrads, they don't really know those opportunities exist. And so I think if they were active participants and actively soliciting, like, I want to be a part of your lab, then absolutely. Now, we all have at the University of Maine something called a capstone research experience, right? And so that is for something like juniors and seniors. So we make sure everybody gets one of those experiences. But I think the president actually is really kind of moving forward and trying to increase that and make sure that all students have what we call an authentic research experience. And I think that's fantastic and really exciting. And so I'm working basically with Scarlett too to try and increase our capacity to have students come and help us on all these really cool projects, because it's helpful to me because we get data, we get this really great data, but it also gives them an opportunity, just like you in a lab, to see if they like it or not. And that is so critical. They don't all need to like it, but what if they do?
This has been really great. I have missed seeing all of my science idols and practitioners. Having an opportunity to talk to you about this, plus your enthusiasm, always makes me happy. So,
thank you so much.
The Maine Science Podcast has received support from the Maine Technology Institute and is recorded at Discovery Studios in Bangor, Maine at the Maine Discovery Museum. The Maine Science Podcast is produced and edited by me, Kate Dickerson. I received production support from Maranda Bouchard, and the Discover Maine theme was composed and performed by Nick Parker.